Dear students, welcome to the sixth session of Histology, Animal Behavior and Applied Zoology paper prescribed by the Mangalore University for 4th semester BSc Zoology. In this session, we shall consider the study of histological features of mammalian testis. As we all know that the mammalian testis is the male sex organ that synthesizes and secretes androgens, as well as it is the site of spermatogenesis. Each male mammal is equipped with two testes, which are generally hung down from a sac called scrotum in order to maintain a bit lesser temperature than the normal body temperature of the mammals, which is very important for normal spermatogenesis to take place. The testes develop in the abdominal cavity and later descend into the scrotum, where they are suspended at the ends of the spermatic cords. They are the sites of spermatogenesis and production of male sex hormones, primarily the testosterone. The outermost covering of the testis is called tunica vaginalis, which is composed of this region is called tunica vaginalis, which is composed of a parietal layer, the outermost layer, and the visceral layer, the innermost layer, and the space in between, which is called cavity. Inside, the testis is covered by a capsule of thick connective tissue called tunica albuginea. This is the tunica albuginea. The tunica albuginea is thick, fibrous connective tissue capsule of the testis. So this forms the capsule of the testis. This is lined by a highly vascular layer of loose connective tissue called tunica vasculosa, which is very important for the blood supply of the tunica albuginea. This tunica albuginea is thickened posteriorly. In this region, it is thickened, as you can see here, to form a structure called mediastinum. So this region is called mediastinum of the testis, from which the incomplete connective tissue septa arise to divide the organ into approximately 250 compartments, which are called lobuli of testes. So you can see here, this is the mediastinum from which septa arise, which divide the entire organ, entire testis, into approximately 250 compartments. And these compartments are called lobuli testis, or lobules of the testis, with more. You can see here the mediastinum of the testis is producing, septa, which forms the lobuli of the testis. These lobuli testis are pyramidal intercommunicating compartments that are separated by incomplete septa. Each lobuli of the testis is composed of 1 to 4 seminiferous tubules. These tubules are embedded in the meshwork of loose connective tissue. These are seminiferous tubules, you can see here. They are highly coiled structures. These tubules are embedded in a meshwork of loose connective tissue containing blood and lymphatic vessels, nerves, and interstitial cells of Leydig. Now let us see what is the importance of interstitial cells of Leydig. The interstitial cells of Leydig are round polygonal cells in the interstitial regions between the seminiferous tubules. So in between the seminiferous tubules we can see certain polygonal cells, these kinds of cells, which are called interstitial cells of Leydig. They possess a large central nucleus, numerous mitochondria, a well-developed Golgi complex, and many lipid droplets. The lipid droplets contains cholesterol esters, which are the precursors of testosterone. Hence, the function of interstitial cells of Leydig is to produce, synthesize, and secrete the male sex hormone testosterone. They are richly supplied with capillaries and lymphatic vessels. The functions of interstitial cells of Leydig. These interstitial cells of Leydig are endocrine cells that produce and secrete testosterone. The secretion of testosterone is stimulated by luteinizing hormone produced in the pituitary gland. These cells mature and begin to secrete during the puberty. So we have seen that each lobuli of the testis is composed of 1 to 4 seminiferous tubules. Let us see what are these seminiferous tubules. Seminiferous tubules are the structures that are 30 to 70 centimeters long with a diameter of 150 to 200 micrometers. They are enveloped by a fibrous connective tissue or tunic composed of several layers of fibroblasts and extensive capillary beds. They form tortuous pathways through the testicular lobules and then narrow into short straight segments, the tubuli recti, which connects with the rete testis. They are lined by a thick complex epithelium, the seminiferous or germinal epithelium. So you can see here the structure is completely lined by a thick complex epithelium, which are called germinal epithelium or seminiferous epithelium. This epithelium consists of 4 to 8 cell layers and contains spermatogenic cells from which the germ cells eventually develop through a process called spermatogenesis, and the Sertoli cells, which have several other functions. See here, you can observe the presence of many layers of the germinal epithelium, and in between you can see long columnar cells, and these cells are called Sertoli cells. Sertoli cells have a pale oval nucleus at the base that displays frequent indentations, they are highly infolded and possess a large nucleolus. They have a well-developed smooth endoplasmic reticulum and some rough endoplasmic reticulum and an abundance of mitochondria, lysosomes, and an extensive Golgi complex. The receptors for the follicular stimulating hormone are also present in the plasma membrane of the Sertoli cells. The Sertoli cells form zonulae occludens, or generally or commonly called tight junctions with adjacent Sertoli cells near their bases, thus dividing the lumen of the seminiferous tubule into a basal and adluminal compartments. So at the base they are connected to one another, so through tight junctions. You can see here superficially they look like they are having no connection, but at the base they have a common connection through tight junctions. So at the base they are called basal surface, and at the above they are called adluminal compartment. These tight junctions are responsible for the blood-testis barrier, which protects the developing sperm cells from the autoimmune reactions. So generally what happens here is the blood will not come in direct contact with the developing spermatozoa. All the nourishment required for the developing spermatozoa will be delivered through the Sertoli cells. What are the functions of Sertoli cells? Sertoli cells support, protect, and nourish the spermatogenic cells. They phagocytose excess of cytoplasm discarded by the maturing spermates. They secrete a fructose-rich fluid into the lumen that nourishes and facilitates the transport of spermatozoa through the seminiferous tubules to the genital ducts. They synthesize androgen binding proteins ABPs under the influence of FSH. ABP assists in maintaining the necessary concentration of testosterone in the seminiferous tubule so that the spermatogenesis can progress. They secrete inhibin, a hormone that inhibits the synthesis and release of FSH by the anterior pituitary. They establish blood-testis barrier. They synthesize and release anti-Mullerian hormone, which determines maleness during the sex determination of the child. For further reading and reference, you can refer these textbooks, which are commonly available in the libraries. Thank you.